For decades, the story of human biology started and ended with a single molecule. We were taught that DNA was an uncompromising blueprint, a hard-coded script that predetermined everything from our eye color to our lifespan. But that rigid view hits a wall when we look at identical twins. Monozygotic twins develop from a single fertilized egg, meaning they are born with the exact same 3 billion base pairs of DNA. Genetically, they are perfect carbon copies. Yet as they age, doctors frequently observe a baffling paradox. One twin might develop severe metabolic disease or cancer, while the other lives completely disease-free. If their underlying genetic code is completely identical, what invisible mechanism is causing their physical bodies to diverge so wildly? The answer lies in a hidden regulatory layer called the epigenome. Translating literally to above genetics, this system acts as the master controller for ourselves. Zooming in on the DNA double helix reveals a multitude of chemical tags physically attached to its outer edges. These compounds do not alter the underlying genetic sequence itself. Instead, they act as switches, turning specific genes on or off. DNA is not a rigid destiny. It is a dynamic, waiting system that requires a separate set of chemical instructions to know exactly how it should express itself. To understand how this hidden layer operates, we have to look closely at the two primary mechanical pathways the epigenome uses to control our genes. The first mechanism is DNA methylation. Proteins attach tiny chemical tags, methyl groups, directly to the DNA bases. These clusters act as a physical mute button. When the cell's machinery tries to read the instructions, the methyl groups physically block the way, keeping the gene silent. The second mechanism is histone modification. Inside our cells, DNA wraps tightly around spool-like proteins called histones. Chemical tags attach directly to them. If instructed to pull tight, genes are hidden. If told to loosen up, the DNA relaxes and unspools, exposing genes for activation. Together, methylation and histone modification act as our biological software. They continuously parse the genetic hardware, deciding second by second which parts of the code are actually utilized. We used to assume these regulatory tags were permanently locked into place during early development. We now know they are highly reactive, constantly being written and rewritten by the outside world. Everyday pressures, our daily diet, chronic stress, and smoking, provoke chemical responses that physically alter our epigenomic layout. The clearest proof of this system comes from moments of severe historical trauma. Researchers studying the survivors of the Swedish famine and the brutal winter siege of Leningrad observed a distinct biological pattern. The extreme starvation experienced by these individuals left durable epigenetic marks on the exact genes responsible for regulating their metabolism. The human body physically records our life experiences and external stresses, etching them directly into our cellular chemistry. In standard biology, there is a strict rule regarding reproduction. When parents pass their genomes to their offspring, the epigenetic slate is supposed to be wiped entirely clean, starting fresh for the next generation. But researchers are now probing a controversial frontier called transgenerational epigenetic inheritance. They are finding that under certain specific conditions, a fraction of these chemical tags manage to survive the reproductive wipe. The implications here are staggering. It suggests that the environmental traumas, famines, and severe habits experienced by an ancestor might leave physical marks that are inherited by their children and grandchildren. This chart overlays complex genetic data onto a multi-generational human family tree, visually representing the search for heritable epigenetic markers. It maps an area of intense scientific debate. While this kind of inheritance has been clearly proven in simple organisms, confirming human inheritance of acquired epigenetic traits is immensely difficult, and researchers continue to scrutinize the data. The genome is not a static archive, but a living, breathing ledger. The environments we inhabit and the choices we make today rewrite our biological software, potentially echoing through the health of generations yet to come.